Hi everyone, welcome back to Dr. Hong's Immunology series. We took a break last week, and this week we'll come back to start our adaptive or acquired immunity module or content. Now we'll begin by talking about cellular immunity. So here are the lecture objectives. So we're going to look at the process of antigen presenting, talk about both MHC class 1, class 2 in greater detail, and also look at cellular immunity and look at how T cells carry its immune response. So the term adaptive immunity basically is interchangeable with the other term, acquired immunity, and depending on which textbook you are reading, you'll see either one of them. Now, this type of immunity is considered as our third line of defense after physical barriers and innate immunity. Now, there are several effector cells, just like our innate immunity, but here we have B cells and T cells, or T lymphocyte or B lymphocyte to be precise. Now, T lymphocyte, in a one word to summarize, it mediates cell-mediated immunity, and B cells or B lymphocytes are responsible for humoral immunity that is based on antibodies. The key features about adaptive immunity is that it is very diverse. All right, diverse in terms of the meaning is that it means it can recognize many different types of antigen, but whenever they recognize, it is very specific, and also they carry memory, meaning that you will remember, or your body is going to remember this particular encountered antigen in the future. Now, in our innate immunity portions, we talked about the concept of self and non-self, and cells in the innate immunity use pattern recognition receptors to tell the differences between human features and pathogen features. Now, this concept of self and non-self also applies in the adaptive immunity, and to tell the differences, we also use specific receptors. So on B cells, we have membrane-bound or surface immunoglobulins, or Ig, that are serving as their receptors. Now, essentially, they are the same structure as antibodies. Look at the figure there. Now, the only difference is antibodies is a soluble form that you can find in your plasma, but membrane-bound receptors that are on the B cells, like the name says, it's on the surface, it's not freely floating. On the T cell side, you also have specific T cell receptors that are on there, and no matter if they are T cell receptors or membrane-bound Ig, they are always very specific for a single target. Now, notice that for a given lymphocyte, so when you're looking at one lymphocyte or one T cells or one B cells, all of them carry multiple of these receptors on their surface. But all of the receptors on a single lymphocyte are specific for the same target. And for a different target, you would have a different lineage of B lymphocytes or T lymphocytes. So this illustration or this graphic actually represents what I just talked about. So you have two different B cells here and you have two different T cells here. Now, they carry different receptors as illustrated with different colors. Each one of them, is specific for a given epitope or a part of the antigens, and their diversity is accomplished at the level of gene, meaning that there are specific mechanisms, we call it gene arrangement, rearrangement, or nucleotide insertions to create a infinite amount of combinations or diversity to generate these different specific receptors. So here we are going to very briefly look at these two mechanisms, and we're not going into details. Now, in terms of gene rearrangements, actually we have more than 130 different alleles in our genes that are available to code for the different variable regions or the antigen binding site on our B cells and T cells receptors. And another mechanism we call nucleotide insertions or deletions can also create additional polymorphisms or differences between the final protein or the receptor products to generate a huge diversity that way. One of those gene rearrangement process, we call it VDJ recombinations. Here we're not going into the biochemistry mechanisms of gene recombinations, but basically here this graph illustrated how different part of the genome, okay, we have various of regions that code for the variable regions of the receptors, and through recombination rearrangement, so you take out some and you added some and generate a different pieces of messenger RNA each time for a different new antigens, and that eventually get translated into the variable regions or antigen binding regions of either the B cell receptors or the T cell receptors. So now let's have a general overview of B cells and T cells development. Now, before they starting to have all these specificity, both of them begin at a stage we call it naive B cells or T cells, which is a cell that has not been activated by a recognizable antigen. Now, after it is being presented with an antigen, following recognitions and activations, that particular lymphocyte will undergo two processes. First is proliferations, meaning to expand the populations that is specific for that given antigens. Then it also starting a process called differentiations, become so-called a armed effector lymphocyte. Now, B cells will become plasma cells, which can actively secrete soluble antibodies, and then eventually become memory cells that carry memories so that the system remember the antigen. Now, T cells can become a helper T cells and also a killer T cells to carry their specific functions, and also become a memory cells to maintain that memory. Now, we're going to look at these specific functions in this lecture and the next lecture. So now we will begin to talk about a process called clonal selections and expansions of lymphocytes. So this process happened to both B lymphocytes and T lymphocytes, but we are using T lymphocytes as illustrations here. So when we face an infection that the innate immunity cannot overcome, the adaptive immunity is going to kick in. So initially, only a very tiny percentage of naive lymphocytes are capable of reacting against any given single part of an antigen. So here we have a graph showing you, 3 naive T cells, okay, and they're being presented with a part of the antigen that is from a given pathogens by a antigen presenting cells. All right, so a small number here I illustrated, maybe 1 of 3, okay, probably would be 1 in many thousands, okay, in reality. So one of them can recognize a little bit, okay, of the pathogen antigens. Okay, now this recognitions, even though it's small, okay, will go through a process called this process is called clonal selections. So only that can really recognize part of the pathogens will get selected. This will ensure specificity. Now, this clonal selection process ensures specificity. It will also serve as the activation stage, so that particular naive T cells are no longer naive, okay? They're activated. Now, after this activations, this single cell begins to undergo changes. Namely, the first one would be proliferations, meaning cell divisions. They're going to expand their number, clonal expansions. So here in the graph, I show that one of them with the red colored receptors expanded from 1 to 3. Okay, now it takes time for cell to divide, so that's why our adaptive immunity takes time. So there's a delayed in reactions. So after it's expanded, it's going to further activate it into and differentiate it into final mature stage into either helper cells, cytotoxic T cells, or memory cells in terms of the T cells. Now, B cells go through a slightly different process, but basically they still go through the clonal selections and clonal expansion. So basically our body clones a lot of these T cells that can recognize, or B cells that can recognize the given antigen. So a little bit humored. So it's just attack of the clones. So I just drew one of those and then copy and paste. So immunology is really like a story, and for adaptive immune response, the story began with antigen presenting. So it requires an antigen presenting cell to start the whole process. It is usually carried by immature dendritic cells and mature macrophages. Monocytes and B cells can also act as an antigen presenting cell, but out of all, dendritic cells really specialized for it. And just like macrophages, dendritic cells reside in the tissues and they can do phagocytosis. Now, the initial recognition is really based on receptor-mediated endocytosis, meaning the dendritic cells is going to eat up the antigens, engulf the pathogens or toxins, depending on what they are. And after the engulfing process, we talked about it in the innate, is going to go through degradations and processing. Basically, that pathogens is going to be broken down into component peptides and carbohydrates and things of that nature, and going to present it back out to the cells in the adaptive branch. So we've talked about this concept briefly in the first one or two lecture in the series. So here is a recap. So here is the process illustrating where are these antigen presenting happening. So all of these antigen presenting cells is going to migrate to local lymph nodes, the so-called the T cell zone. Now, after the antigens are being processed, it's going to display on the surface and presented to a series of naive T lymphocytes. And within the cortex of the lymph node, naive T lymphocytes are going to get screened for the specific binding or any type of a binding for that antigen. So, and then go through the process. We just talked about the clonal selections and clonal expansion. Now, once the antigen-presenting cells, such as dendritic cells, taking up the antigens and being broken down, it's going to load onto the MHC molecules, which are then being exported to the surface of the antigen-presenting cell. Now, using T cell again as an example, here that is going to make brief contacts with the antigen-presenting cells and allowing the interactions between the T cell receptors, the MHC complex, and also in between those is a peptide antigen from the pathogen. Now, if the T cell receptor does not bind well to the peptide, that cell-to-cell contact is going to break off. But if it binds well to that given peptide from the pathogen, the contact is going to be sustained, and then T cell is going to be activated through the process clonal selection and expansion. So that was the overview of antigen presenting to T cells. So where are all these different antigens, or what could be the possible pathogens? Now, mostly here in this course, we are going to look at bacterial and viral antigens. So bacterial generally they are considered as extracellular antigens. So the bacteria usually thrive on the surface of human epithelial cells. Now, there are specific or different bacteria that resides within a cell. So that is not all the case, but a lot of the time they are extracellular. And bacterial antigens are usually encountered in these extracellular space and is processed by phagocytic cells and being presented to those T cells and B cells. Now, on the other hand, viruses are always inside our cells. They want to thrive and replicate inside of our cells, and their antigens are usually encountered and processed intracellularly within the infected cells. And however, there are cases that complete viruses or viral particle may also be encountered in extracellular extracellular space and being processed by phagocytic cells. This can be happen, but most of the time they are within our infected cells. So these are the different sources of antigens. The reason we talk about different sources of antigens because these dictate a slightly different pathway of how our lymphocytes are being activated, and we're going to look at the specific mechanisms in the next slide. So we had an overview of the antigen presenting process in the previous few slides, and basically it can divide it into two basic routes, okay, and these two basic routes is through two different classes of MHC molecules. And first we're going to look at MHC class 2. Now, MHC class 2 processing or presenting is involved in presenting antigens from extracellular space or surface or compartment. Usually we're talking about bacteria or intact virions that are being encountered outside of the cells. Now these extracellular antigens are going to be internalized and being engulfed by antigen presenting cells into vesicles, and the vesicle is going to fuse with the lysosome and being broken down by proteases and hydrolases within the phagolysosome, and those proteins being degraded into small peptide pieces, and these peptide pieces will bind to MHC class 2 molecule and being presented on the surface, so forming this MHC class 2 peptide complex that are on the surface of the APC. So and then you wonder, where are these MHC class 2 coming from? Now it's originated from the Golgi, okay, and then it's going to fuse with those vesicles that's containing different peptides, and that binding will happen, and then that vesicle is going to transport to the surface of the antigen presenting cell. The second route of antigen presenting is for presenting antigens that are within the cell, internal cellular compartment. So this process is through MHC class 1 molecule. So the figure here illustrates intracellular antigens, it could be the productions of a viral proteins, during the viral replication process. Now these proteins will be broken down by our internal mechanisms called proteosomes, These proteosomes will chop up these viral proteins, chop up a little bit, not all of them, and part of these or pieces of those peptides is going to being transported to the endoplasmic reticulums at the same time, and then the Golgi will also have this MHC class 1 molecule being transported and fused into this vesicle, and those peptides is going to bound to the MHC class 1 molecule and transport to the surface of any cells. Any cells meaning here is any infected cells by any cells that are infected by the virus. So the figure here illustrates how the MHC class 1 molecule being expressed on the cell surface. So the picture here really trying to illustrate the difference between MHC class 1 and 2 is that you can imagine there are two attaching points, to the cell membrane, and then MHC class 1 having one attaching point on the cell membrane. So that would be the graphical differences of those two classes of molecule. So now we have a little understanding of how antigens or peptide antigens are being processed and present on the surface of antigen presenting cells. And this lecture we're going to focus more on T cells. So here again we're looking at how T cells are going to interact with antigen presenting cells. Now this antigen presenting cell and T cells interactions, the cell-cell interaction, we call it immunological synapse, where there is this communications happened after this contact. Now look at the picture here. It is quite a complicated setup. So we have MHC class 1 molecule or bound to a peptide antigens and interact with T cell receptors that is on a type of T cells, we call it CD8, and we have MHC class 2 molecule bound to a peptide antigens and interact with T cell receptors that are on a different subclasses of T cells, we call it CD4 T cells. Now, what are these CD? Now, CD represents a cluster of differentiations. Okay, now these CDs are actually markers on T cell surface that are co-expressed with the receptors and act as a co-receptors. And these co-receptors are necessary for successful cell-cell contact. Now, for CD8, they are usually presented on all of the cytotoxic killer cells or cytotoxic killer T cells. And CD4, they are presenting on all of the helper T cells. And so this graph illustrated the different combinations there. Now, and then you ask me, how should I remember these CD4, CD8, and MHC class 1, MHC class 2? It's very confusing. Now, there is one easy way to remember this. Okay, now we have CD8 pair with MHC class 1. Okay, so 1 and 8, so 1 times 8 equals 8. Okay, hold that thought there. And we have CD4 match with MHC class 2. 2 and 4 together. 2 times 4 equals 8. And 1 times 8 equals 8. So with that acronym, I think you should be able to match very well. CD8 match with MHC class 1 and CD4 match with MHC class 2. Now we will begin to talk of cellular or cell-mediated immunity. Now, cell-mediated immunity refers to the branch of the adaptive immune response that is carried out by T cells. Now, the first major type of T cell is CD4 T Helper Cells. Now, T Helper cells, they all have CD4 as their marker. Now, CD4 T helper cells can secrete different cytokines that can activate other cells, such as phagocytes, macrophages or neutrophils, and as well as activate B cells. Now, helper T cells respond to peptide antigens that is presented by MHC class 2 proteins that is on the surface of the antigen presenting cells. So that means they are engaged in responding to extracellular antigen. The second major type of T cells are these CD8 killer T cells or cytotoxic T cells. Now, cytotoxic T cells, they are responsible for destroying cells that are infected with intracellular pathogens. Mostly we're talking about cells that are infected by viruses. Now, they can react against cells that bear antigenic peptide that is presented by MHC class 1 proteins and potentially located on any nucleated cells. So these CD8 T cells are engaging intracellular antigens. So here is another look of how intracellular antigens are being processed. So here is a graph or illustration showing you a virus being infecting any cell types, and during their viral cycle, their viral proteins are being translated. And within our cells, the protective mechanisms called proteasomes, they're going to degrade part of these viral proteins and being presented with the MHC class 1, okay, on the surface of the cells. Now remember, all these presented antigens are in peptide forms, okay, very important. They're peptide forms, and they're going to present it to cytotoxic T cells, and once they're recognized, these abnormal peptides that are not from within the body, they're going to do the killing action. Now let's take a deeper, detailed look of how killer T cells recognize MHC class 1 and the antigen that is presenting. So the figure here showing you an antigen presenting cell that is presenting a piece of antigenic peptide on the MHC class 1 molecule, and that molecule and the MHC class 1 is going to interact directly binding to a T cell receptors. Here is on a naive T cell, so this is showing some type of interaction. So this is during the early phase, okay, early screening phase before the clonal expansions, okay. Now important to note that CD8 co-receptors, are there present. Now, in addition to that cluster of interactions, there are also other co-receptors that are very important, that are absolutely required for successful T cell activations, which are the B7 marker and CD28 marker. Now notice here I have a bracket there. B7 marker is a marker on antigen presenting cell, and CD28 marker is present on the T cells. These interactions are absolutely required for successful T cell activation following the recognition between the MHC class 1 peptide and T cell receptor. After the successful T cell activations following recognition, the activated naive T cells is going to secrete IL-2. Now this IL-2 is going to self-activate, induce proliferations and differentiations, and develop into effector functions that can carry actual cytotoxic activity. So activated cytotoxic T cells is going to recognize the same type of peptide that are present on an infected cells and perform the killing action. So how does cytotoxic CD8 T cells carry out its effector functions, meaning how does it destroy virally infected cells? Now, it's going to release a array of compounds such as cytokines and cytotoxic granules. We've seen some of those, right? Perforins. Now, perforins is going to poke a hole in the target cell membrane. And granzyme is going to initiate apoptosis, aka programmed cell death. A new molecule here is the Fas ligand. A Fas ligand is going to released by cytotoxic T cells and bind to a Fas receptor that is located on the infected cells and also initiate apoptosis via something called caspase enzyme. Now, in addition to that, cytotoxic T cell is also going to release interferon gamma, which is a type 2 interferon that also directly inhibits viral replications through interrupting protein translation process, and also induce more MHC class 1 expressions, and also as well as stimulating phagocytic cells. And last but not least are different TNF alpha and TNF beta. Now we have a different name to collectively call TNF, which is a lymphotoxin. Now the functions of TNF alpha and beta is to activate more macrophages and also killing some cells via binding to TNF receptor, perhaps through apoptosis mechanism. So you wonder, how do T cells find out which cells are infected and which cells are not infected? In fact, there are even more molecules that are interacting between T cells or cytotoxic T cells and their target cells before the killing actions. And these molecules, we call them adhesion molecules. And here are some of these adhesion molecules that we've seen it before. We've seen these same adhesion molecules when we talk about neutrophils, right? So we have LFA1 and ICAMs. So these adhesion molecules allow cell-to-cell contact. Now the LFA1 is also called integrin. Integrin. They're expressed on the T cell surface. And correspondingly, ICAM or CAM, these intracellular adhesion molecules, are expressed on the target cell surface. Now, if there is some type of peptide that are presented on the infected cells, these non-self peptides are being presented, and this adhesion molecule after contact will induce cytoskeleton rearrangements to facilitate T cells to release these cytotoxic granules and perform those killing actions that we just talked about. So when we look at the mechanism behind these cell-cell contact and cell adhesions, it actually is quite interesting. And then you look at this figure, what am I talking about? What is Dr. Hong referring to? All right. So in fact, these cell-cell contact is not so much different than meeting someone and dating. And let's look at why. All right. So the initial contact between CD8 and potential target cells was not through T-cell receptors or MHC. They actually occur through these adhesion molecules, namely the LFA1 integrin and ICAM. So initial contact between these adhesion molecules are relatively non-specific. It could happen to anyone. And when there's these initial contact happen and there's no MHC with foreign peptide, there's no sticking and no killing. Now, when there is second contact, okay, if there is a MHC molecule bearing foreign peptide that is recognizable by the T-cell receptor, and magic will happen and chemistry will happen. It's going to rearrange these cytoskeletons and induce degranulations and kill the target cells. So that's what I said. It's almost like meeting someone. Initially, if you meet someone, it could be very casual. You could say hi to anybody, right? Very non-specific. But if that person have something that you are looking for, namely these, peptide, that some chemistry, you're gonna have some tight binding. You may go on a date and some chemistry happen, and eventually you may just, come out of it, just present some type of flowers or gift and realize, like, can we just, be together, right? So, and, degranulation, that is a degranulation process, essentially. All right, so that's why I said these T cells and target cells interaction is very much like just dating, and that's why we have these pictures there. Now, I purposefully draw these figures as gender neutral, so just in case. All right, after we look at the MHC class 1 pathway and how it process and present foreign peptide or intracellular antigens to cytotoxic T cells or CD8 cytotoxic T cells, we are going to look at how MHC class 2 present its antigen derived from extracellular infections to helper CD4 T cells. So the basic mechanisms of antigen processing are pretty similar between the different pathway. So the extracellular antigen is going to be engulfed by antigen presenting cells and broken down into peptide pieces and presented via MHC class 2 on the antigen presenting cell surface to CD4 T cells. Now CD4 T cells, they are helper T cells, and just like we have naive CD8 T cells, we also have naive CD4 helper T cells, and they are usually designated with something called TH0. Now, in fact, helper T cells can develop into at least two, even more. Okay, one of the at least two subtypes. So the major one that we are talking about here is developing into TH1 helper cells and TH2 helper T cells. Now, this is because from, got stimulated from different cytokines environments, and they can actually have different responses. So I have a big picture here. TH1, okay, is responsible for helping or stimulating macrophages, and TH2 helper T cells is responsible for stimulating B cells. And there's actually a very fine balance between TH1 and TH2, and it is believed that any imbalance between TH1 and TH2 helper T cells may play a significant role in the pathology associated with allergies and other autoimmune disorders. So just to jog your memory a little bit more, right, here is the graphical illustrations of TH0, meaning like naive T helper cell. No, naive, they have no expressions, and they can differentiate into either TH1 or TH2. So I purposefully draw their eyes with numbers just to tell you the difference. All right, again, the big pictures here is that Th1 helper T cells have a role in activating macrophages. So it's going to have phagocyte activations and also help to induce destruction of intravesicular pathogens, meaning those that are already engulfed within the macrophages. So how does it help? How does it induce more activations of macrophages? It is through all these different cytokines that is being released, namely there are TNF alpha, interferon gamma, and as well as interleukin 2. Now, interleukin 2 has a role to further expand the populations of Th1, right? And interferon gamma can at the same time induce the or activate the macrophages, can also have an effect on inhibiting Th2 growth, which is on the humoral or antibody responses. So it shows you, you are activating one branch, okay, you're enhancing more Th1 mechanisms, you are suppressing Th2 mechanisms at the same time. So Th1 helper mechanisms, we also have a dedicated name called cellular helper, okay, cellular helper, because it is helping cellular actions. So within this balance, the Th2 is helping humoral responses. So when you hear humoral, you immediately want to link it to B cells, okay, antibodies. So when you have, a dominated Th2 activations or Th2 inductions there, it will help to induce more B cell proliferations and differentiations into plasma cells, and then further down the road to actively secrete soluble antibodies. Now, it can be achieved by different cytokines as well. First is interleukin 4. Now, interleukin 4 secreted by Th2 helper cell is going to promote B cells and plasma cells and antibodies there, so it's dedicated with the green arrow. And IL-4 at the same time can also inhibit Th1 growth. So here we see again the same theme. When you have more Th2 development there, you inhibit Th1. So there's a balance there. Now, other molecules that are also play an important role would be the IL-10. Now, IL-10 has a role in inhibiting Th1 growth as well as inhibiting also the macrophage activation cellular response. So here is the the balance between Th1 and Th2 helper cells. So putting the two pieces of information together, together in one page. So Th1, it is really important to remember that it facilitates phagocytic activities and promotes destructions of intravascular pathogens. And Th2, it activates B cells and promotes plasma cell secretions of soluble antibodies. And this balance is achieved by different cytokines that are released by either Th1 or Th2. And oftentimes when you have a cytokines that can stimulate, the Th1 branch would have inhibitions on the Th2 branch and vice versa. So immunology, again, is always like a story. Now we had a big picture, and then we focused on the smaller pieces here. Okay, what is the detail? Okay, how or what actions actually happening when Th1 cells activates macrophage? Now, when it's activated, the phagosomes within the macrophages will more efficiently fuse with lysosome, forming phagolysosome, and that would help to facilitate a faster breakdown of these intravascular pathogens. And it will also increase the productions of reactive oxygen species and proteases. So those are the compounds to help breaking down these pathogens. And most importantly, the TNF alpha and interferon gamma will also increase MHC expressions and increase B cell markers on the macrophages. So what do they do? Well, they have a role in antigen presenting. Remember, macrophages can do antigen presenting. So they after they break down these intravascular pathogens is going to be more efficiently present those pieces to further activate other T cells, more likely the naive T cell that can do cytotoxic activities. So earlier we looked at how adhesion molecules facilitate cell-cell interaction between cytotoxic T cells and target cells. And the same situations or same mechanisms also happen with helper T cells and their target cells. Now, instead of a cells that is going to be killed by T cells, or cytotoxic T cells, T helper cells, target cells, are macrophages, macrophages, for examples, like here particularly for Th1 helper cells. Now, similar mechanisms also happen with Th2 helper cells and B lymphocytes. So we have the same molecules having the integrins that is located on the Th cells and interact with ICAM that is on the macrophage here on the graph here. Now these interactions are usually, again, very loose. It's not specific. But if a macrophage is presenting a particular antigens that is matching with the T cell receptors that is on the Th helper cells, is going to have the higher affinity binding and trigger more stronger formations between the integrin and ICAM. And this prolonged cell-to-cell contact is going to allow subsequent cytokine release and activate the macrophage. So here is the summary of some of the most important information in this lecture, which is the MHC class molecules and the different pathways of how they present peptide antigens. Remember, they are peptides. Now, for cytosolic pathogens, namely those that are infecting the cells, also we talked about viruses. Now, the proteins is going to be degraded within the cytosol by proteasomes, and they're going to bind to MHC class 1, which is present on all of those nucleated cells. And MHC class 1 is always going to present a peptide to T cells that carry CD8 marker. After its activations, the effector CD8 cytotoxic T cells is going to also synapse on effector cells and going to induce cell death. Now, on the other side here, we also look at intravascular. So, antigens or pathogens that are being engulfed by professional antigen presenting cells and going to be inside a vesicle. Now these vesicles eventually is going to filled with lysosome, forming these phagolysosomes and being degraded. The degraded pieces of peptide is going to be presented on MHC class 2 molecules. Now remember, these are located only on professional antigen presenting cells. Now these class 2 molecules with the peptide is going to synapses on T cells that carry CD4 marker, and this presentation is going to activate the cells. So here again is another review and summary of what we just talked about in a word format. So that is all for today's lecture. Now this lecture we focused on cellular immunity and how it interacts with peptide. So I keep saying peptide because next lecture we're going to look at humoral immunity, and humoral immunity we're referring to B lymphocytes. Now there's going to have some tie to this lecture, but at the same time there are some significant differences between how B cell receptors, namely membrane-bound immunoglobulins, recognize their antigens versus T cells recognize their antigens. So stay tuned for the next lecture, and I'll see you next time. Bye.